Okay, guys, you're welcome to IGCSE Biology syllabus content 20.3, and here we'll be looking at genetic engineering. And quickly, this is our syllabus content. This is all the concepts we need to know for both core and extended. So, this is what we are going to cover in this episode. So, quickly, let's move. Genetic engineering, what does it mean? So, genetic engineering simply means changing the genetic material of an organism by removing, changing, or inserting individual genes. So genetic engineering simply means changing the genetic material of an organism by removing, changing, or inserting individual genes. Now, example of genetic engineering include the inserting of human genes into bacteria to produce human insulin. So, the inserting of human genes into bacteria to produce human insulin, and also the insertion of genes into crops to confer resistance to herbicides, the insertion of genes into crops to confer resistance to insect pests, and also the insertion of genes into crops to provide additional vitamins. So these are examples of genetic engineering where you have um, um, crops that are herbicide resistance, and you have crops that are pest resistance. You have genetically modified crops that have additional vitamins and the production of human insulin. All these are examples of um, genetic engineering. So genetic engineering is the process also of taking a gene. So look at this. It's a process of taking a gene from one species and putting it into another species. So we'll look at that now shortly. So you see, the control of all the normal activities of a bacterium depend upon its single chromosomes and small ring of genes called plasmids. Now, genetic engineering will not be possible in the absence of plasmids. So plasmid help genetic engineering to be possible. Now, in genetic engineering, pieces of chromosomes from a different organism can be inserted into a plasmid. These allow the bacteria to make a new substance. Now, there are some ethical concerns about biotechnology. So, and the ethics of genetic engineering benefit. It helps to cure for diseases such as cryptic fibrosis and cancer. Food, which is healthier in the case of genetically modified crops, and stay fresh for a longer period and taste better. But risk of genetic engineering is that unknown effects of moving genes from one organism to another. New dangerous disease are being created, and it's usually against God and nature. So, what are the application of genetic engineering? Application of genetic engineering, it's more like what I've explained earlier in this part. This example is similar. So, application of genetic engineering in the production of human insulin. So, insulin, this hormone can be produced by genetically modified bacteria and has been used since 1982. And the human insulin gene is inserted into bacteria, which then secrete human insulin. We'll look at how that process takes place because it usually comes out as 6-mark questions. And in the case of pest resistance, the bacteria called Bacillus, oh God, damn, thuringiensis, just know it, try and pronounce it. It's quite difficult for me. Produces a toxin that kill caterpillars and other insect larvae. Now, this toxin has been used for some years as an insecticide. So, genetic modified crops. So, genetic engineering has huge potential benefits in agriculture, but apart from a relatively small range of crop plants, most developments are in the experimental or trial stage. In the USA, 50% of soya bean crop and 30% of the maize harvest consist of genetically modified plants, which are resistant to herbicides and insect pests. So herbicide resistant. So some of the safest and most effective herbicides are those such as glyphosate, and which kill any green plant but become harmless as soon as they reach the soil. So these are ways in which genetic engineering have been applied in recent years. Now, using genetic engineering to put human insulin gene into bacteria. So this is the aspect I was saying earlier that comes out as 6 mark question. It's quite straightforward. You just need to understand it. You need to understand the concept. You need to know how it works. Now, the first thing you need to know is using human insulin gene into bacteria. So first, there are 2 organisms involved. We have the human cell, human insulin cell, and a bacterium. So every bacterium, this now a human cell, and this is a bacterium. And inside the bacterium, they contain plasmid. So remember, I said plasmid is the useful organelle in the bacterium that help in genetic engineering. So this the gene coded for production of human insulin can be isolated and using restriction enzymes, and also help to form sticky ends. So also the plasmid is isolated from the bacteria using restriction enzymes. So the gene for the manufacture of particular insulin is now inserted um, into the plasmid, which is cut with the restriction enzymes. Now, using a ligase enzyme, so ligase is used to join them together. Now, once that is done, the plasmid is now inserted back into the bacteria, and it keeps reproducing to produce more insulin. So this is the process majorly. Now, first, isolation of DNA making up human gene using restriction enzyme, and also forming sticky ends. So you isolate the DNA for making insulin, this human gene called insulin, using restriction enzyme from the human cell. Now, cutting of bacterial plasmid DNA, so the bacterial plasmid is also cut out from the bacterium with the same restriction enzyme, forming complementary sticky ends. So find out that this plasmid that has been cut has complementary ends, sticky ends to this insulin DNA from humans. Now, insertion of the human DNA into bacterial plasmid DNA here using ligase enzyme to form recombinant plasmids. Now, these are recombinant plasmid that is formed now. So, insertion of the plasmid into the bacteria back. So, this recombinant plasmid is now inserted back into the bacteria. So, replication of bacteria containing recombinant plasmid, which make human protein as they express in the gene. So, this once it reproduces in a higher quantity, can now be isolated and be used in the process. Now, this diagram here try to explain the same thing. So, see, this is a human insulin gene, that code for that helps in producing insulin. So, this part is the insulin part. So, it is cut off using a restriction enzyme and forms sticky end. So, the bacterium plasmid is also cut using restriction enzyme. So, plasmid cut open with restriction enzyme. Same thing here. And gene cut out with restriction enzymes. So both of them will now have complementary sticky ends. So the ligase enzyme stick insulin gene into plasmid, just like this. Now, which form recombinant plasmid. Plasmid is now put into a bacteria, like this. Then bacterium with insulin gene grow in a fermenter, reproduce in larger quantity. So the insulin is now separated off and purified. And you remember, insulin is used by diabetic patients to convert excess glucose into glycogen for storage. Now, lastly, the syllabus expects us to know how genetically modified crops are, the advantages and disadvantages. Now, first, from the explanation so far, and genetically modified crops are pest resistant, they are herbicide resistant. should know that first. And all this, it is uniform in shape, so it's easy to transport, and it's appeal to customers because of how beautiful it looks. Growing season is usually shorter. It has a shorter growing season, and most of them are drought. They also have ability to form drought resistant variety which need less water. It gives higher yield, and is also pest resistant. But the disadvantage of genetically modified crops are natural species of this crop may die out. Taste is often not good enough as the natural species. It leads to development of superweed that are stronger than the genetically modified crops itself. And no one knows the long-term effects of genetically modified crops on humans. So with this, we are done with this topic on biotechnology. So subscribe so you get notification once I upload the next